Good evening. Do you hear me well?
Yes.
Okay. Okay, so I first would like to thank the organizing committee for inviting me here to this nice meeting to talk about telomeres and ovarian aging. And just to start, I will say that when we are born, we have a few years of health, but then at the age of 45 to 50, all these, the incidence of cancer, diabetes, and all these aging-associated diseases start. And at the same time, in the case of women, menopause start, causing infertility and increasing the risk of aging-associated disorders. So one of the pathways which is involved primarily in aging is the telomere pathway, not only because telomeres shortens as we age, but also because it is related to other pathways that also impact aging, such as genome instability, stem cell exhaustion, cellular senescence, mitochondrial dysfunction, epigenetic alterations that we are gonna explain a little bit about. So here we have an image of telomere fish. Telomeres are those yellow dots at the ends of chromosomes. They are nucleoprotein structures which protect chromosomes and help the cell distinguish between the natural ends of chromosomes and double-strand breaks, which must be repaired. So telomeres protect from chromosome fusions and degradation activities. In mammals, telomeres consist of tandem repeats of the TTA-GGG sequence. And telomere length is already established depending on the species. For instance, in humans, telomeres are about 10 to 15 kilobases, whereas in mice, they are about 40 kilobases. Telomeres end in a single-stranded DNA overhang, which is able to invade the double-strand DNA to generate what is called the telomere loop. The telomere loop is helping hiding the very end of chromosomes from the rest of the nucleus of the cell. Telomere repeats are bound by a complex of proteins which contains six subunits called sheltering, whose main function is protection, just to help maintain chromosomes safely, avoiding the activation of DNA damage signals. The sheltering complex binds DNA through the TRF1 and TRF2 subunits, and they help stabilize the telomere loop. They also can recruit telomerase. They can help with telomere replication, and they control telomere length. But telomere structure is a little more complex because histones have epigenetic marks. They have methylation marks, which generate an atmosphere that is refractory for gene transcription. And this has an effect already on genes which are downstream telomeres, is the telomere position effect. And on the top of this, telomeres are bound by long non-coding RNAs called TERAs, which also prevent telomere lengthening or unscheduled telomere recombination and help hide telomeres from the nucleus of the cell. Because of the unidirectional nature of telomere replication, the replication machinery, the very ends of chromosomes cannot be copied, and this makes telomeres shorten upon cell division. When telomeres reach a critically short length, then the DNA damage response pathways are activated. If these signals are persistent, then the cell cycle arrests and cells go into senescence or apoptosis. And this is very important because if this happens to adult stem cells whose function is to replenish tissues, the tissue homeostasis, if they enter senescence, they could not divide further to regenerate tissues. And this is also causing aging. And this can happen if one cell has like 4 or 5 telomeres, which are critically short. But if many telomeres, for instance, 20, 30 are critically short, then cells enter crisis. There is gross chromosomal rearrangements, chromosome fusions, and genome instability, which may lead to tumor formation or tumor development. So the telomere shortening is solved by the telomerase. Telomerase is a reverse transcriptase, which is composed of a protein component and an RNA component. The RNA component is used as a template for telomerase to add de novo telomere repeats onto chromosome ends. So basically, telomerase could maintain telomeres, could prevent fast telomere shortening, but telomerase is not active in most cells of the body. It is only active in cancer cells, in embryonic and adult stem cells, and in the germline. Here you can see that telomerase is very active in immature oocytes, and as oocytes mature, telomerase activity decreases. It stays not so active during cleavage stage embryos. Then at the modular stage, it increases, reaching a peak at the blastocyst stage. And something similar is observed with telomeres. Telomeres are longer in the immature oocytes. As oocytes mature, telomeres shorten. And also during cleavage stage embryos, telomeres remain short. And then at the morula to blastocyst transition, telomeres lengthen. So when I was in Maria Blasco's lab, we found that the telomeres of the inner cell mass of the blastocyst, which is going to give rise to the embryo, are the longest telomeres in the blastocyst, longer than the trophectoderm. But what are the consequences of telomere shortening? When telomeres shorten, we can see at the third generation without telomerase, mice look very old. They have a lot of white hairs compared to controls. Telomeres shorten, and in some cases here, in some chromosomes, telomeres cannot be detected by FISH, by in situ hybridization, sorry. The regenerative capacity of tissues is very decreased due to stem cell exhaustion, and the size of the ovaries is also very reduced, although it seems that they contain all sorts of, all types of follicles. So the size and shape of the ovary and the uterus is very variable, but very weird in these mice at the 6th generation without telomerase. The weight of the ovary is half of it should be due to this problem of tissue homeostasis, and the function is also altered. So they produce less oocytes. When they are fertilized, some of them reach the modular stage, but most of them don't even reach the blastocyst stage. And when oocytes already at the 4th generation are analyzed, what we find is chromosome misalignment and spindle aberration, which for sure difficult embryo development. And this might explain why at later generations, they are infertile. But furthermore, these mice have shorter lifespan. So a wild type can have a mean lifespan of about 2.5 years. And in successive generations without telomerase, lifespan is decreased. And at the 3rd generation without telomerase, these mice already look old at the age of 1 year. where they should just be adult, but not old. The advantage, if there is any, is that these mice are cancer resistant. So in humans, something similar happened. We are born with long telomeres, 10 to 15 kilobases. In somatic and even in adult stem cells, telomeres shorten with aging. If these telomeres get critically short, cancer may be developed. But in humans with mutations in telomerase gene, this telomere shortening is accelerated, and they have all these problems. Their regenerative capacity of tissues is very limited, so their organs fail and their lifespan is shorter. And actually they develop what is called the Telomeric Syndromes. These are very severe diseases such as dyskeratosis congenita, aplastic anemia, idiopathic pulmonary fibrosis. So these are really bad diseases, and this telomere shortening, of course, is also affected by lifestyle choices. Like smoking is not good, the perceived stress, and unhealthy lifestyle, like sedentary habits. So telomere length in individuals is very variable for a given age. Some people will have longer telomeres than the mean. Other people will have shorter telomeres than the mean. But the people with shorter telomeres will have an increased risk of suffering aging-associated diseases. And this is the reason why telomeres are bona fide markers of biological age, not chronological age. So recently, there have been many publications, or several publications, relating telomere length with infertility. In fact, in the ovary, some somatic cells, like granulosa cells and cumulus cells, have long telomeres and telomerase activity. This is important because they have to divide intensively to nurture the oocyte as it matures. And what these papers found is that the telomere length and telomerase activity of granulosa cells of patients with primary ovarian insufficiency are null, or short telomeres, or null telomerase activity. So the telomere pathway is really affected in the cases of infertility. In addition, people who carry the BRCA1 and 2 mutations in combination with short telomeres have a higher risk of developing breast and ovarian cancer. So there is this relationship, but one of the main questions that we have is whether this aging can be reverted or not. So one way to revert aging and rejuvenate tissues and organs would be through telomere lengthening. So all you know that differentiated cells can be rejuvenated just by transduction of the introduction of the 4 Yamanaka factor, CMYC, OCT4, SOX2, KLF4, and then cells become induced pluripotent stem cells. So basically, this is what I show in the picture. With 2 factors, it was not possible to generate these pluripotent cells, but with 3 or 4 factors, these cells were generated. They are able to create any tissue, any cell from the organism. But what happens to telomeres when cells are rejuvenated? Here in this picture on the left side, I show the telomeres of differentiated cells. They are short, no more than 50 kilobases, well, normal for mice cells. On the right side, telomeres from embryonic stem cells, which are much longer, about 100 or even longer than 100 kilobases. So when differentiated cells are induced to pluripotency with 3 or 4 factors, telomeres lengthen, and lengthened until the point to reach the telomere length of embryonic stem cells, which are really very, very long. And all this happens by telomerase reactivation. In fact, here, differentiated cells have short telomeres, like it was shown previously, and in telomerase deficient mice, the cells can be reprogrammed, but telomeres don't lengthen, and this reprogramming actually is at a very, very low efficiency, and it doesn't have the same potential as when telomeres are longer. So before I continue showing you things, I want to explain you how we measure telomeres. So in order to do this, we do first in situ hybridization. We use a telomeric probe. It contains 3 telomeric repeats, and it's labeled in one end with CY3. So we hybridize the probe with chromatin, and then we make intensive washes to remove the non-specific binding. And then we acquire C-stacks images in a confocal microscope. We make a maximal projection, and then we use the Definition software to analyze images. If images are really very big, the software just divided in small parts. It first finds every nucleus that we have in the picture, and then it finds every telomeric spots, and the information is exported. We can have intensities from telomeres or cells. We can export information the way we want. It's a very versatile program, and we obtain it in data in Excel documents, or we can create these maps where we have the cells with the longest telomeres, which are the red cells here, right? So when I was with Maria Blasco, we found that the telomeres of the inner cell mass were very long, but not so long as the telomeres of embryonic stem cells. So we wonder whether these long telomeres of embryonic stem cells would be inherited from the inner cell mass of the blastocyst or not. And in order to understand this, we extracted blastocysts from mice. We cultured them in vitro in conditions where the trophactoderm is destroyed. It will not grow, only the inner cell mass will grow. Then we passage these cells onto 96 well plates. Here's where the embryonic stem cells start to emerge. And upon successive passages, these cell lines are established. And what we found is that the in vitro inner cell mass, the inner cell mass culture, had really very short telomeres compared to the in vitro inner cell mass of the blastocyst. And telomeres lengthened upon in vitro expansion this in a similar way as the induced pluripotent cells would make it. So basically, these very long telomeres are not inherited. They just, telomeres are lengthened. So we took embryonic stem cells expressing GFP with very long telomeres, introduced them in morula, allow them to reach the blastocyst stage, and then what we found is that the telomeres at the blastocyst stage in cells expressing GFP were still very long. So we wondered if we could generate mice with very long telomeres and how would they behave in vivo. To this purpose, we use embryonic stem cells at passage 4, which have normal telomeres, and at passage 16, which had very long telomeres. And then we analyze different tissues at up to 1 year of age. So I have to say that we had 2 different backgrounds here, the 129, which had the very long telomeres or normal telomeres, and the VL6, which were the recipient morula. And also the 129 background with long or normal telomeres would generate these dark patches that we could see on chimeric mice. So we first analyzed the content of GFP cells in different tissues of these mice, and we saw that they were retained up to 12 months, they were still there. And we did this, we used this telomapping technique, and we could see that the places where the GFP was expressed, telomeres were very long compared to the places where in this case, the parts of the intestine where the GFP was not expressed. We also saw that the adult stem cells, which in the intestine case is the crypt, had longer telomeres compared to normal cells. And also the villi, the differentiated compartments, had longer telomeres. So we wanted to know whether these telomeres behave normally, and we can say, yes, they behave normally. They just shorten with age as normal telomeres do, but over time, they tended to accumulate less percentage of critically short telomeres compared to normal cells. We did analyze the levels of TRF1 sheltering and found that they were well protected. We also analyzed damage looking at the gamma H2X, which marks double strand breaks, and we saw that over time, they accumulated less damage than normal cells. So here in the image, the GFP expressing cells don't show a spot as normal cells, but we still needed to know whether these telomeres suppose were an advantage for the organism or not. And so we did wound healing experiments. We used circular razor blades of 3 millimeters. We caused wounds and we followed healing. And we could see here in the 129 patch on the first row, the wounds at day 7 were already healed compared to any other cells, which were just controlled and had shorter telomeres. So somehow telomeres, long telomeres give some kind of advantage. But the other way of rejuvenating organs and tissues could be telomerase reactivation. So the first time that telomerase was overexpressed, it was done from birth. Tissues overexpressing telomerase healed faster, had better regenerative capacity, but there was an initial death caused by spontaneous tumor formation. So it was difficult to know whether mice would live longer and better, and to circumvent this problem, the super mice was generated. These mice would overexpress telomerase in a cancer-resistant background, meaning that they would express 3 tumor suppressors. So you can see here in the image that at the 2 years of age, these mice look younger compared with wild type mice. Here we did telomapping with skin, and you can see in the lower images that the super mice have more cells with very long telomeres, even at older ages compared to controls. They also wonder whether, because we not only want to live longer, but we want to live better. So we want to be healthy for longer time. This means cognitive function, cardiovascular status, bone density, even metabolic fitness. But I'm going to show you only this neuromuscular coordination test. Here mice are asked to walk around the bar, and they can do it 5 times. And if they do it 3 times without falling, the test is considered positive. And while wild type mice at older ages would not do this test properly, the super M mice did it really well, independently of the age. More importantly, these mice live longer, 40% more, even 50% if the cancer cases were not taken into account, and the health span was much longer. And this is very important. If we translated this to humans, we could say that our maximum lifespan would not change, but the mean lifespan would be 115 years. We would be healthy for about 85 to 90 years, and then we would decay very fast. But unfortunately, humans are not cancer resistant, so other strategies had to be explored. In this case, gene therapy with adeno-associated viruses expressing telomerase gene or just GFP. They were introduced when mice had 1 or 2 years old. They were adult or old. Here in the Western blot, in the lower part, we can see that telomerase was detected when the virus contained the telomerase gene and not the GFP. These viruses can actually infect many tissues. They would be able to have a sustained gene expression even for years, and they are non-integrative. So basically, after telomerase injection, here, these graphs show the accumulation of short telomeres, and at older ages, at 2 years age, it is more visible that the amount of short telomeres is decreased. And actually, these mice also perform better these experiments, neuromuscular coordination. Actually, it was much better when the gene therapy had happened for 11 months compared to 6 months. And more importantly, these mice live longer. Particularly, well, in the older group, even the maximum lifespan had been increased. And these mice didn't show any more cancer incidence compared to a control. And this is very important. This could actually be used in humans. This doesn't seem to create more cancer, and it extends lifespan and health span. And just to almost finish, I want to show you this other experiment where telomerase gene was fused to the estrogen receptor and in an inducible cassette, so that if tamoxifen was not added, telomerase would not refold properly, it would be inactive. But when tamoxifen was added in the drinking water, then telomerase would be reactivated. So this group generated, they went to the 4th generation of mice without telomerase, and they found all these symptoms that telomerase deficient mice have. In fact, here we can see that tissues were atrophic. They found in spleen and intestine signals of DNA damage, but upon telomerase reactivation, the signals of damage disappeared. This is quantified here. Apoptotic cells also decrease. The spleen tissue, spleen weight gained weight. And this also happened in the case of testes. They gained weight and they were functional. The PAPs per liter were increased. Actually, in this work, the authors explain that probably even in atrophic tissue, some adult stem cells are quiescent. And when telomerase is reactivated, these cells can start to divide and replenish tissues. So with this, and just to finish, I want to say that perhaps we can do interventions, telomerase reactivation or others, when people are still healthy, just to try to increase the years in which we are still without diseases and shorter the aging period, and even perhaps avoid having some of these aging-associated diseases. And hopefully we would manage also to extend the reproductive lifespan. And actually in the laboratory, we are working in some projects trying to rejuvenate organs and understand telomeres, ovarian aging from the telomere point of view. And so with this, I would like to thank IVIRMA founders, presidents, and directors, because they push research a lot in the clinics. And we have all this atmosphere with doctors, biologists, embryologists. So it's a very interesting place to be working. And I want to thank the funding agencies. And also I want to thank all of you for your attention. And I am open to questions.
Dear Dr. Varela, thank you very much for this really very interesting presentation and this very sophisticated research on telomere activity and function. Really congratulations for your research. And now it's a turn of audience to ask some questions to you.
Mm-hmm. Thank you.
There is There is no questions. So, okay, maybe it will be interesting for us just to give some information. Is it possible, according to you, to use stem cell therapy for ovarian insufficiency and somehow to reactivate telomerase activity for treatment of this condition?
In fact, yeah. Stem cells have been used to rejuvenate ovaries, and actually it's very interesting because they can wake some of the dormant follicles, and this is very interesting. But I mean, some colleagues at IBIRMA are doing this kind of job, but unfortunately I haven't been able to analyze telomere length on these samples. This is actually one of the things that I am hoping to do at some point, because I find it very interesting. Yeah.
So it's a hope for the future, and maybe how to balance also the risk effect of telomerase activity. But I hope it will be the treatment of the future. Thank you very much again.
Thank you. Thank you.